چیز شد نه Hello everybody. I am Dr. Hossein Gudarzi. I am head of Microbiology Department of Medicine Faculty. My degree is full professor. And I'm so happy for your servicing about microbiology and virology. I hope you would be cool, safe, and happy everywhere and forever. Microbiology is a kind of medical sciences who studies about microorganisms and needs to some microscopes, just as bright field microscope, electron microscope, dark field microscope, confocal microscope, and immunofluorescence microscope. All of them can help us for observation of microorganisms, bacteria. There is some highlights about microbiology and bacteriology. In Old Testaments and documented about Old Testaments, there is some documents about leper disease, that it is a contagious disease. And some highlights about some scientists in microbiology there is. For example, Varro described for the first time principle of contagion about microorganisms. And next, Roger Bacon told these microorganisms are living creature. After
Excuse me, sir.
Yes.
We cannot see the PowerPoint presentation.
Yes. Of course, maybe cut off PowerPoint, and if it would be cut off, you must send your email or you write my email and I will send for you. Roger Bacon told these microorganisms are living creature. After Roger Bacon, Fra Castorius was the first person who told these living creatures are Seminaria morbi and they are contagious disease. After Fra Castorius, Antony van Leeuwenhoek was the first person who observed microorganisms. Of course, he used a microscope by his own design. It was a kind of glasses and some lenses. He observed a drop of water and he observed some microorganisms. Of course, he didn't know what is them. After Van Leeuwenhoek, Robert Hooke combined microscopes and told cellular theory about microorganisms. After him, Carl von Linné told these microorganisms have genus and species and divided microorganism to genus and species. After Linne, John Snow in England understood some epidemic disease and microorganisms. Edward Jenner was the first person who described variolation or vaccination. After Edward Jenner, Louis Pasteur and Spallanzani told that living creature is not spontaneously. They performed demonstrate that microbial life does not arise spontaneously, and there is some way for entering of microorganisms. For example, water, for example, airborne and soil. After Pasteur and Spallanzani theory, Pasteur described and proved alcoholic fermentation, and he made some vaccine, for example, anthrax vaccine, rabies vaccine, and chicken chlora vaccine. After Pasteur, two Joseph Lister and Semmelweis used some disinfectant and phenolic spray in operation room, and they control and decrease some morbidity and mortality rate in patient in operation room. After them, Ivanovsky was the first person who described viruses. And after Ivanovsky, Robert Koch was the first person who cultured microorganisms on the culture media, on the agar. And two microorganisms, Mycobacterium tuberculosis and Bacillus anthracis, grows on the culture media. In 1928, Fleming discovered penicillin. In 1935, Waxman discovered sulfamide. And in 1945, Rodenberg discovered electron microscope and bigger magnification for microorganisms. And finally, Mullis in 1983 discovered PCR, or polymerase chain reaction. That is a good technique for detection and diagnosis of infectious disease microorganisms with a good sensitivity and specificity, and is a rapid test. This is a brief history about microbiology that I told you. Now, Robert Koch conclusively showed that microbes can cause disease. We have two kinds of cells. Some of them are eukaryote and another are prokaryotes. Eukaryotes are divided three groups: animals, plants, and protista. Animals, plants, and protista. And protista divided four groups: algae or unicellular or multicellular, protozoa or unicellular, fungi unicellular or multicellular, and helminths or multicellular parasites. They are protists. And now, of course, added slime molds, a kind of fungi, to these groups. But it is not our mean. Our focus is based on prokaryote. The size of prokaryote is 1 micrometer, about micrometer, 0.5 to 2 micrometer. It's average size. These groups have no intracellular organisms, just as endoplasmic reticulum, Golgi apparatus, and their ribosome is 70S or sedimentation rate. In contrast to eukaryote, their sedimentation rate is 80S. It's important because some antibiotics and some mechanism effect related to these ribosomes. Bacteria belonged to prokaryotes. Their cytoplasm is amorphous and some primitive enzymes there is in this cytoplasm. This group have no real and true membrane on the nucleus. Their nucleus or genomic matter is nucleoid. Prokaryote divided three groups: archaebacteria or ancient bacteria, archaebacteria or eubacteria. All of the pathogenic bacteria are in eubacteria. And finally, cyanobacteria or bluish and green cyanobacteria or alga, that their structure is near to prokaryotes. But our means and our focus is based on eubacteria. For identifying of bacteria, we have some criteria or some taxonomy. The first taxonomy is appearance of bacteria. For example, some bacteria have spore, some bacteria capsule, and figure out of some bacteria, for example, Cucus, Bacilli, Vibrio, and Spirochetes. This is morphology of bacteria or appearance of bacteria. This is the first criteria. The second criteria that is so useful for detection and has good sensitivity and staining, Gram staining. Christian Gram was a Danish scientist who described Gram staining for differentiation between some microorganisms. The mechanism of Gram staining depends on the thickness of cell wall or peptidoglycan, the number of pores on the cell wall, and the permeability of peptidoglycan or cell wall. For that you will do in our laboratory, you must fix your specimen on the slide. For fixation of bacteria, we have chemical and physical methods. In physical method, we use from thermal ways. And in chemical, we use alcohol methylic or methylic alcohol for fixation of bacteria. Of course, in our laboratory, you will use from thermal path. After fixation of bacteria by thermal ways, you will add a kind of color, violet d'eau gentian. All of the bacteria take this color, and all of them would be violet.
Excuse me, sir. Your slides are not visible.
My sound?
Sir, your slides are not visible.
Yes, no. I will see. I will show to you. Please notice to my speak. Some of bacteria are gram positive and some are gram negative. Gram positive
Excuse me,
are
sir.
yes.
So sorry to interrupt, but a lot of the students from our batch are not able to join the meeting.
Yes, yes. Gram staining needs to fixation of bacteria. For fixation, you have two pathways. Chemical, in physical, you have thermal pathway, and in chemical, you use methylic alcohol. After fixation, you will add violet de Janssen. All of the bacteria will take violet de Janssen and will be violet or dark bluish. After washing, you must add Logol or gram iodine. Logol or gram iodine. It stabilizes violet Janssen in bacterial cell wall. After 3 minutes, you must decolorize these colors by using of alcohol and acetone, about 15 to 30 seconds. In this step, you have two groups of bacteria. One of them are colorless and one of them remain their color. And finally, you need to counter staining or adding safranin and fuchsin. By adding safranin and fuchsin, colorless bacteria will be red or pink, and it has no effect on bluish or violet bacteria. Conclusion is, gram-positive bacteria are dark bluish or violet, and gram-negative bacteria are red or pink. It is important for the first observation of bacteria. It's a good diagnosis of some microorganism, but it is not specific. You must notice that golden test in microbiology is culture. Golden test is culture, and gram staining and other staining only helps you for better diagnosis. Of course, we have two exceptions. One about syphilis disease, Treponema pallidum, because this microorganism is so dangerous. You will use serology tests and other molecular tests for diagnosis. And Mycobacterium leprae, the cause of leper disease, because its generation time is so long, and it needs to about 6 months. I will repeat, we have two exceptions for culture: syphilis disease, Treponema pallidum, and leper disease, Mycobacterium. The third criteria for taxonomy and identification of bacteria is metabolic pathway. For example, catalase and oxidase test. Catalase test is important for differentiation between Staphylococcus and Streptococcus. Streptococcus is catalase negative, and Staphylococcus are catalase positive, or some bacteria, for example, Vibrio cholera and Pseudomonas aeruginosa are positive oxidase. It is important in primitive diagnosis. The fourth pathway and criteria for identifying of bacteria is extracellular enzyme production and pigmentation. For example, some bacteria, just as Pseudomonas aeruginosa, can produce piocyanin that have some color, for example, yellow, green, red, black, and brown. It helps for us for diagnosis in Pseudomonas aeruginosa infections. The fifth way and criteria is genomic pattern. Genomic pattern. It is the best way for diagnosis of microorganisms and bacteria. It's based on hybridization of DNA and percentage of adenine and thymine, guanine and cytosine. It uses in molecular methods for rapid diagnosis of bacteria. The sixth criteria is phage typing. It is important in nosocomial infection in hospitals. Phage is bacteriophage or viruses that is significant and specific for each bacteria and may be important for pathogenesis of some disease, for example, in diphtheria. And finally, the last phylogenetic drafts. Phylogenetic drafts. Based on phylogenetic drafts, we have 4 types of bacteria. First, Gracilicutes or gram-negative bacteria. Gracilicutes or gram-negative bacteria. Second, Firmicutes or gram-positive bacteria. Firmicutes or gram-positive bacteria. Third, Tenericutes or Mulicutes or non-cell wall bacteria, just as Mycoplasma. And fourth, Archaeobacteria or ancient bacteria that have no cell wall or peptidoglycan. I will repeat: phylogenetic draft criteria: Gracilicutes or gram-negative, Firmicutes or gram-positive, Tenericutes or Mulicutes in non-cell wall bacteria, just as Mycoplasma, and Mendozicutes or Archaeobacteria or ancient bacteria. It was a simple criteria for dividing and taxonomy of bacteria. In this slide, you will see size of bacteria and viruses. One of the smallest viruses, poliomyelite virus, about 27 nanometer. And the size of bacteria, you will see E. coli and rickettsia. And in comparison to red blood cells, the size of bacteria is about micrometer, and size of viruses is about nanometer. Bacteria that has known until yet is nanobacter, that its size is about 200 nanometer. And the biggest bacteria that has known yet is Q. margarita namibiensis. Q. margarita namibiensis. That it's about 1 millimeter, the size of bacteria. The smallest is nanobacter, and the biggest is Q. margarita nami. You will show the comparison between prokaryote and eukaryote, and some character of prokaryote and eukaryote, just as fungi and protozoa. In prokaryote, you will have circular DNA molecule not covered with protein or not cellular membrane. But in eukaryote, you have complex of DNA and basic proteins and just particles, for example, endoplasmic reticulum, Golgi apparatus, and the ribosome is about 80S or sedimentation rate. Of course, in prokaryote, the ribosome is 70S ribosome. About localization of nuclear structure, in prokaryote, it's dense tangle of DNA in cytoplasm and no nuclear membrane, I told you, or nucleoid or nuclear equivalent. Of course, in eukaryote, in nucleus surrounded by nuclear membrane or real nuclear membrane. DNA of prokaryote is nucleoid and plasmids and sometimes transposons and SXT elements and integron. Of course, in most of time, you will have nucleoid and plasmids. But in eukaryote, in nucleus and in mitochondria. About cytoplasm, the cytoplasm of prokaryotes is amorphous, I told you, and you have no organelles just as mitochondria and endoplasmic reticulum, and ribosome is 70S. But in eukaryote, you have mitochondria and endoplasmic reticulum and ribosome 80S. About cell wall, in prokaryote, you have rigid cell wall with murine layer or peptidoglycan. Of course, exception is mycoplasma that has no cell wall. And in eukaryote, present only in fungi, glucans, mannans, chitin, chitosan, and cellulose. I told eukaryote just as fungi, protozoa, alga, and some slime molds. And about reproduction, in prokaryotes, reproduction asexual or non-mitotic by binary transverse fission or binary fission. But in eukaryote, in most cases, sexual, possibly asexual. You will show eukaryote cells and the organelle of the cytoplasm, Golgi apparatus, ribosome 80S, rough endoplasmic reticulum, lysosome, nuclear membrane, endoplasmic reticulum, cell membrane, and mitochondria for respiratory of eukaryotes. And in contrast, in prokaryote, you will see cell wall or the important section of cell wall, peptidoglycan, and the gene is single supercoiled circular chromosome. Single supercoiled circular chromosome. The length of DNA is about 1 millimeter, but it impacted by 2 helix and superhelix configuration, and it has been 1,000 less, about micrometer. And its weight is about 2 or 3% of cellular bacteria, and the volume is about 10% of cells. And you will see plasmid or extra chromosomal DNA and cell membrane. It is so important. It is for your examination, cell membrane, because it is the only vein for respiration of bacteria, just as mitochondria in eukaryote cells. And cytoplasm is 70S ribosome, and it has 2 fractions, 30S and 50S. They are important in mechanism effect of antibiotics and resistant to them. The structure of microorganism is very simple, but their envelope is very complicated in contrast to eukaryotes. You will show you and you will see gram-positive and gram-negative envelope. In gram-positive cell wall, you will see a thick layer of peptidoglycan in outer and plasma membrane in inner layer. And between our peptidoglycan, you will see some teichoic acid or teichoronic acid. Teichoic acid is a polymer of ribitol or glycerol, and it is specific for gram-positive bacteria and has antigenic effect. Teichoic acid and some polysaccharides, just as rhamnose, arabinose, galactose, glucosamine, glucuronic acid, and manuronic acid are some polysaccharides. The function of teichoic acid is stabilizing of cell wall. The function of teichoic acid or teichoronic acid is stabilizing of cell wall. In contrast of gram-positive, in gram-negative cell wall, we have no thick peptidoglycan. We have a thin peptidoglycan. This peptidoglycan is between outer membrane and inner membrane. In gram-negative, you have two differences: thin layer of peptidoglycan and outer membrane, OM. Our conclusion is this, that we have two differences between envelope of gram-positive and gram-negative. In gram-positive, we have teichoic acid or teichoronic acid and thick peptidoglycan, and in gram-negative, we have thin peptidoglycan and periplasmic space between inner and outer membrane. And also we have outer membrane. Outer membrane is specific for gram-negative bacteria. You will see gram-positive bacterium, some organelles, just as nucleus without any membrane, 7S ribosome. In some bacteria, you will have capsules. Cell wall, plasma membrane. S layer is the first layer of bacteria that can attach to epithelial cell. And in some bacteria, for motility, you will have flagellum. Plasma membrane structure. It is bilayer phospholipids. All of the bacteria have plasma membrane or cytoplasmic membrane or inner membrane. And this cytoplasmic membrane has no estrol in all of the bacteria. Only exception is Mycoplasma. Mycoplasma is the only bacteria has no estrol or has only estrol or cholesterol. All of the bacteria has no estrol. Exception is Mycoplasma. The duties of the cell membrane is transfer of electrolytes and food and balancing of osmotic pressure for bacteria. Second, delegation in peptidoglycan synthesis by lipid carrier. Third, respiration of bacteria just as mitochondria in eukaryotes. It is so important for respiration of bacteria. Four, secretion and excretion of some exoenzymes, for example, protease, amylase, and pectinase for breaking off some molecule for using a bacterium. And finally, chemotaxis effect is one of the duties of cell membrane. I will repeat the duties of cell membrane. It is bilayer phospholipid. All of the bacteria have no estrol in their cell membrane. Exception is mycoplasma. Their duties is 1, electrolyte and food transfer. 2nd, delegation in synthesis. 3rd, respiration. 4th, secretion of enzymes. And 5th is chemotaxis effect. It is electron graph of typical gram-positive cell, and you will see peptidoglycan and thick peptidoglycan. You will see inside peptidoglycan teichoic acid and teichoronic acid. I told you it has antigenic effect, and it is important for gram-positive bacteria. And from inner to outer, you have plasma membrane and then peptidoglycan. This is the structure of teichoic acids. It's a polymer of ribitol and glycerol, I told you. And in contrast, the gram-negative envelope, you will have periplasmic space with a thin layer of peptidoglycan, and you will have plasma membrane or inner membrane and outer membrane. In gram-positive, we have no outer membrane, but in gram-negative, we have outer membrane. In segment or a part of outer membrane, we have lipopolysaccharide or LPS. LPS is so important for gram-negative bacteria. It has 3 segments: lateral polysaccharide or O antigen, lateral polysaccharide or O antigen, core polysaccharide, and KDO or ketonic acid. It's an inner sugar in lipopolysaccharide. And finally, A-lipid or lipid A. It's disaccharide combined to fatty acid. A-lipid is so important for gram-negative pathogenesis because it has endotoxin effect. It cannot secrete outer of bacteria. It's a part of structure of bacteria, and it's thermostable. And the genus of this endotoxin is lipid. In contrast of endotoxin in bacteria, we have exotoxins. Exotoxin excrete and secrete by gram-positive and gram-negative bacteria. It is thermolabile and sensitive to thermal. And the genus is protein. I repeat again, endotoxin is specific for gram-negative, but exotoxin excrete and secreted by gram-negative and gram-positive bacteria. Endotoxin of bacteria have some biological effect that I will tell you. First, hypotensive shock. Hypotensive shock, because it decreases cardiac output. It decreases cardiac output and causes hypovolemic shock. Hypovolemic shock. Second, a complication or DIC, intravascular coagulation, designated intravascular coagulation. And finally, causes hemorrhage from vessels. Third, positive Schwarzman test. When you inoculate endotoxin to rabbit, it causes toxic endotoxicosis and necrosis. Fourth, increase of tumor necrosis factor and cachectin. 5, increase of alternative pathway complement activation and release of anaphylatoxin and chemotoxins. 6, hypoglycemia due to glycolysis. 7, leukopenia and compensated leukocytosis. 8, release of endorphin and enkephalin that causes euphoria in some patients after infection by gram-negative bacteria. It was a bio effect of endotoxins. Of course, about exotoxin, the number of bacteria and the level of exotoxin is important for pathogenicity of an organism. Another section and segment of bacteria in structure is capsule. Capsule has gelatin layer, and the genus of capsule may be different in some bacteria. For example, in Streptococcus pyogenes, the genus of capsule is hyaluronic acid. And in Bacillus anthracis and Yersinia pestis, the genus of capsule is protein. And in Klebsiella pneumoniae and pneumocoque, the genus of capsule is polysaccharide. Capsule in gram-negative bacteria is so important, or K antigen. It is protecting layer for bacteria, and it has some determinant or receptor for some bacteriophage. Other is flagellum or flagellin. It has composed from some protein just as flagellin. It is H antigen for gram-negative bacteria. It related to motile bacteria, for example, in Proteus, in Clostridium. We have four types of flagellum or four types of filaments. One monotrichous, for example, Vibrio cholera, the cause of cholera, monotrichous flagellum. The second is amphitrichous or biflagellum. For example, in Campylobacter and Helicobacter. The third flagellum is Lophotrichus. For example, in Pseudomonas aeruginosa infections. And the last, Peritrichus, in Enterobacteriaceae, Peritrichus, that causes motility of bacteria. Flagella is important for gram-negative bacteria because it's H antigen. Our conclusion is that we have three kinds of antigen in gram-negative of bacteria. O antigen or lateral polysaccharide, K antigen or capsule, and H antigen or flagellum of bacteria that causes of motility of bacteria. And there is only in motile bacteria. This is immunofluorescence staining of bacteria. You will see bacterial capsules, and electron graph you will see the size of gelatin layer of our capsule. Other section of structure of bacteria is bacterial pili or fembaria. Bacterial pili or fembaria. We have two kinds of pilus or bacterial pili. One is cellus for genomic conversion, and other pili or common pili. We have three types of common pili: adhesins, agresin, and evasin. They are important in pathogenicity of bacteria and important for vaccine synthesis in some bacteria, for example, in Neisseria gonorrhoeae. The size of pili is shorter than flagellum. You will see bacterial flagellum, bacterial. And in this slide, you will see flagellar motility. Some motility, for example, forward run, tumbling in some bacteria. Other structure of bacteria is spore or endospore. It is a resistant form of bacteria. We have two kinds of bacteria: vegetative form and endospore form. When some situation use chemical agents, for example, antiseptics, or decrease of some water and increase of thermal situation, bacteria goes to endospore formation. Spore have some segments. It's important for your examination. In the central of spore, you will see core or genome of cell wall, core or genome of cell wall. And sometimes you have some dipicolinic acid that is a kind of salt, and some cysteine that is resistant to radiation. After cell wall, you have cell, after core wall of spore, you will have cell wall. That cell wall composed from peptidoglycan. After cell wall, you will see cortex that is so thick, and also it has peptidoglycan, but it's looser than cell wall. After cortex, you will see spore coat, the thickest section of the spore, and is the cause of resistance to antibiotics because it has keratin-like section. And finally, you will see the outer of spore exosporium, that it is a disaccharide and some fatty acids. This is another image of spore. You will see DNA, ribosome, glycolytic enzymes, cytoplasmic membrane, spore wall, and normal peptidoglycan. Cortex that I told you, it's a thick layer of less cross-linked peptidoglycan, and keratin spore coat that is resistant to antibiotics. Spore can survive adverse condition for years. We have some kind of endospore spore formation. For example, in A image, you will see central spore, B is subterminal spore, C is terminal spore, and D is terminal spore with swollen sporangium. This is the steps of endospore formation. And this is a typical classification of bacteria. In gram-positive bacteria, we have cocci and cocobacilli. For example, in cocci, you have Staphylococcus aureus, Streptococcus pyogenes, Enterococcus, and in Coccobacilli, Brucella, Francisella, and Yersinia. In gram-negative, you will see cocci and rods. Cocci just as Neisseria, Moraxella, Kingella, and rods just as Enterobacteriaceae, for example, E. coli or Escherichia coli, Klebsiella, Enterobacteria, Serratia, and Salmonella. And also some other Pseudomonas and Strophomonas, Stenotrophomonas maltophilia, and Proteobacter. And some bacteria are aerobic and other are anaerobic. For example, of anaerobic are Clostridium, Bacteroides, Peptostreptococcus, and Actinomycosis. And atypical just as Mycoplasma, Chlamydia, Rickettsia, and Legionella. Until now, if you have any question, please. Question about classification, about structure of bacteria.
No, sir.
Yes?
No, sir.
No, sir. Yes, very good. How many person in this your classroom?
125.
Your what?
125.
125. Thank you so much.
Yes, sir.
When you will go back? Go back to Iran. Never?
So we will be there by a second week of October.
Oh, yes. Thank you so much. And if you want, you can send your email. For example, it's better your representative classroom or not my email, hgod500 or Hossein Gudarzi 500 @gmail.com. Maybe please write it again. hgod 500 @yahoo.com or Hossein HOSSEIN Gudarzi GOUDARZI @gmail.com. That I will send for you PowerPoints and PDF files for more studies of you. If you have any question, I am at your service, please. If you have no question, thank you so much for your attention, and I hope see you as soon as possible in Tehran. Bye, everybody. Thank you so much. I like everybody. Bye, my friends.
Bye, sir.
Thank you. Thank you, my dear.